first thing we can learn from super resolution microscopy is whether how two proteins are organized spatially and whether they share some interaction space. And I brought you this example, which is it's coming to age. It's a T cell that has been infected with HIV. And we have two proteins labeled. The first protein is the F protein, the other one the GAG protein. And conventional microscopy, or let's call it diffraction limited microscopy, suggested a co-localization of these two proteins. Now we switch on the super resolution mode and we see that the picture changes. We see that the virus, the GAG protein, the lattice is in the middle, and it's surrounded by the F protein. And the F protein has a very high density, so that really changes the understanding of how this protein is, how this virus is assembled. And we can go beyond, and this is a special feature of these single molecule super resolution methods. We can get numbers. And you've seen how these single molecule methods work. So we highlight single fluorescent dyes, proteins, and we collect a lot of these coordinates and put together an image. And we can use this data to quantify proteins. And for that, I have an example. In prokaryotes, this is an E. coli cell, a tiny, tiny E. coli cell, where we labeled the RNA polymerase. And not only that we can visualize these tiny clusters, we can also count how many proteins are in there. And E. coli is a great organism because it's relatively simple to modify proteins and introduce a fluorescent protein for that. And we can do that in a correlated effort together with the chromosome or track these proteins in the live cell. But just to have us all on the same page, we are using single molecule localization microscopy, and Marcus has introduced this technology. What we basically do is we not record fluorescence image at once in one shot with a camera or scanner. Instead, we collect a sort of a huge stack of images, thousands or 10,000s of images. And in those images, we make sure that we only detect single fluorophores. And there's a magic which makes us able to collect these single fluorescent dyes, which is basically shortly summarized here on the right. And this magic is either photochemistry or some photophysical properties or some sort of affinity properties of the probes. I'll not go into these details. Just to cut it very short, we can either work with fluorescent proteins that can be photoactivated, and by using appropriate laser light, we can achieve the single molecule density, or we can use organic dyes which we photo switch, or we can use labels which reversibly bind to their target, and if they do so, they're stuck. We collect the fluorescent signal, and then they dissociate when the signals go. And we record these movies, and we can generate these beautiful super resolution images. So much about that. Please keep it in the back of your mind. And now I'll go to the membrane proteins. And there again, this one is disconnected. Oh, that's the worst which actually can happen. Microsoft PowerPoint has stopped working. Well, in that case, let's restart. That's okay, I'll give it a try. So my apologies, I have no idea. There we are again. Let's see how long that lasts. Okay, I hope so. Yeah, it is. Okay, so receptor proteins. We have heard about, or we have seen beautiful structures about receptors. So we are good at the angstrom level, cryo-EM, X-ray, whatsoever. We are also good at the other scale, or the other edge of the scale in biology, which is we can observe how these proteins behave in cells, live cells, and so on. But we have this gap here in the middle and in between, which is the sort of resolution gap, which we can fill or which we can close with super resolution microscopy. And by that, it's possible to visualize individual receptor complexes. We can learn how these receptor complexes constitute into functional units and initiate signaling. And that's the first thing, and quite interesting stuff you can do with that at the functional level. You can, for example, in this example, TNF receptor 1, you can try to understand how the receptor is associated before it binds a ligand, which is important because it's chemistry. It's the number of interactions which governs the biochemical reaction at the end. And it's also important because biochemistry has produced different results. So using different cross-linkers, some groups reported that this receptor is dimeric, others said it's trimeric before activation. So obviously, or maybe the cross-linking reagent has an influence. It's a quite drastic process to extract a membrane protein. That's one thing we can study. And the second thing, we can add a ligand, and then we can look what happens with these proteins that associate to this receptor. And TNF receptor is a very interesting one because it can induce two very different responses, which is cell death or NF-kappa B activation, proliferation. And somehow this is related to these membrane-bound receptor hubs which form. So it's a very functional state. Now I promised you super resolution will close again, but there's one thing I didn't say. We have a resolution in the range of 10 to 20 nanometers, but this is not sufficient to sort of resolve the number of units within a receptor. So we need an additional tweak on super resolution microscopy. And this is what I want to introduce at the beginning, and let me do that with an example. The prerequisite is, first of all, that we label our receptor with a stoichiometric fluorophore. Simple version or simple solution is use fluorescent proteins, generate a stable cell line. We have to be careful with expression levels, look at the functionality of the cell line, and then do super resolution imaging. For TNF receptor, we constructed a MEF cell line which carries MEOS2. That's one of the favorite fluorescent proteins in the lab. It's green before it gets photoactivated, and then we put a little bit of UV light there, and then we get these beautiful little spots, and from that we reconstruct a super resolved image. Let's look inside. This is the conventional image, and this is the super resolved one. So we can see these clusters, these protein clusters. We don't have the resolution to decide whether this is a monomeric or a dimeric protein. But not all is lost. We have additional information. If we look very closely into these movies, we'll find that these fluorophores, other than it's shown here down in the bottom, they don't light up once, emit light, and then they are gone. Instead, they light up multiple times, something we call blinking. And this is inherent to most fluorophores. It's a pain on the one hand, but you can make use of it. And in this case, it's a benefit. Because this reaction, this sort of blinking, is governed by physical chemistry. And this is proportional to the number of molecules which are within this isolated spot. And then this tells us whether we have 1, 2, or 3 receptors. To summarize this, before I explain you the physical chemistry background in 2 slides, what we will use is super resolution images. We look into individual clusters. We use the additional information of this blinking stuff, which comes for free, and then we determine whether we have monomers or dimers or trimers. Now there are the 2 slides for physical chemistry. So anyone who's not happy with that, don't worry, because afterwards I come back to the biology and I'll just use this technology. Now the background, as I explained, is basically kinetics. So what we need is a model for fluorescent proteins, which beforehand is not activated, non-fluorescent. Then we sort of add some activation light that can be UV light. The fluorescent protein fluoresces, and it might go into this non-fluorescent dark state, which is something we observe as blinking, shown here on top. And this reversible transition into this blinking state competes with bleaching, with this photodestruction. The most simple analysis is to count the number of events which occur until the fluorescent protein dies. To histogram that and to put that into a model. And this model equation comes directly from this kinetic scheme. There's one value, which is this p-value, the probability that the fluorescent protein does not blink, and it incorporates these 3 rates, because these 3 rates govern how many times the fluorescent protein blinks. So this is the explanation. It becomes a little bit more complicated if we have more than 1 fluorescent protein, because then we have to incorporate that these proteins might not be detected. So there's a stochastic parameter which comes in, which is a binomial distribution. But at the end, we end up with equations which can describe monomers, dimers, trimers, and so on. And I should mention that we got help from Gerhard Ruhr from the MPI in Frankfurt. And then we tested these. First of all, we used reference proteins. And the first one is CTLA4. That's, since yesterday, a quite famous protein, because it was Allison who discovered it first in '95. And this protein is a dimer. So we imaged this protein fused to the MEOS2 protein in the HeLa cell. We recorded the blinking distribution. We can fit it very well, and we extract, indeed, it's a dimer to 100%. Well, 1 is not enough, so we went through a couple of more proteins, from monomeric proteins like CD86 and others, through a trimeric protein, VSVG, and to other distributions of proteins, CD80, which appears to be monomeric and dimeric at the same time. This one result on the right is a quite important one, because it shows that we can deal with heterogenic distributions, mixtures of monomers and dimers. Now back to the biology. First of all, TNF receptor 1, where it all started. Let's go back to these questions I asked you earlier. Whether the resting receptor is monomeric, dimeric, or trimeric. So now we know how we design our experiment, our stable cell line, integrated, MES2. We record these images, you've seen that before. But now on top, we analyze the blinking patterns of each of these spots. And then this gives us the answer that TNF receptor 1 is monomeric and dimeric, apparently in equilibrium, about 2-to-1 ratio. There are no trimers at all. That's interesting. So we looked a little bit into biochemistry, and TNFR1 is a very well-studied system. And there's a domain here on top of this receptor which is called the pre-ligand assembly domain, the PLAT domain. And it's apparently this PLAT domain which drives this weak dimerization. So if there's a domain which drives something, we can mutate it out. We did 2 different mutations. I can't see them, but hopefully you can see them. And when we did so, we don't see any dimers anymore. This is good news. We played around with drugs. I'll only mention one to show the potential of the method. It's a drug called sapphirocast, which actually is just known since about a year. It was John Sykes who reported that recently. Since about a year, that it also inhibits TNFR1 activation. So we added this drug, and also this drug drives this equilibrium towards the monomers. So this was the first part. Now we can add the ligand. Now we do a multicolor or a 2-color experiment. So we added a ligand which carries a fluorophore, and we need to do that before we analyze the cells. Because you see here, this is a very tiny part of the cell. It's quite heterogeneous. Some receptors carry a ligand, others don't. Now we can use this 2-color signal and put this sort of information, these clusters, in 2 baskets. The one with and the other one without ligand. For the one without ligand, we get a very similar ratio like we get for the resting receptor. So it seems to be receptors not being activated. For the ones carrying a ligand, we get largely a proportion of trimers, about 2/3. We still have some monomers and dimers, which gave us a hard time, but we now know that TNF-alpha also binds to monomers and dimers with a much weaker affinity, so that fits. And we also get higher order oligomers. Those are quite interesting. I wouldn't speculate too much about the stoichiometry. There seem to be some hexamers, and there are some ideas how these could be organized. But to cut that short, we now have a method that we can sort of visualize and quantify protein complexes with sort of molecular resolution in the cellular context. TNF receptor 1 is a relatively simple system. Speaking, it has only one ligand, one known ligand. There are other receptors which can bind many ligands. Now I come very briefly to a second example. And the second example is toll-like receptor 4, which we happily studied with our colleagues in Reading, Darius Videra and Graham Cottrell. Brief introduction again. TLR4 is a pattern recognition receptor, and it's targeted by lipopolysaccharides, which are excreted or secreted by bacteria. And then it starts a signaling cascade, actually 2 different ones, and it can either activate NF-kappa B again, or ERF3. And the interesting question is, why are polysaccharides act differently on the receptor? And how is this related to the organization of the receptor? And there is some history behind that, some publications suggesting that this receptor organizes into large clusters, whereas other, mostly biochemical methods, suggested that there would be monomers and the activated receptor would be a dimer. So perfect target for us. So we studied that using the very same method. We began with a suitable cell model again, very important. No random cell or random overexpression. That's really not good with these receptors. And again, we used MES2. And the first thing we did is sort of a sanity check. We know from biochemistry that there are 2 co-receptors. The one is MD2 and the other one is CD14. If we remove them, the receptor is dead like a doornail. So we essentially generated a cell line which doesn't have these 2 co-receptors, and we compared that data to the one which carries the 2 receptors. And what we find is, for the native case of the resting TLR4, we find again a ratio of about 1-to-1 of monomeric and dimeric receptor. So there seems to be an equilibrium again of those dimers with the monomers. If we remove these co-receptors, we only have monomers. And in that case, biochemistry shows us that this receptor is not signaling anymore. So this was our sanity check. So apparently, we have to start again from a 1-to-1, or at least from a certain portion of dimerized, pre-dimerized receptor. And please don't take these numbers too serious. This is the result of our method, but there's some variation in biology. Right? Okay, now the next step was to test different ligands, LPS. And we started with our resting cell, and we added 3 different species. The one species were derived either from E. coli and the other one from Salmonella, and both showed that it drives the receptor towards more dimers. And it's apparently those dimers which bind to the downstream signaling proteins and start NF-kappa B nerve-free signaling. The third species we used was Rhodobacter. Rhodobacter LPS turns the whole receptor monomeric. No dimer at all, so it's very antagonistic. And apparently at the receptor level, it already stops any kind of signaling with a very different organization of the hub, of the central hub of receptor activation. And we can put that into a model, in a very simple model, of how the receptor organization changes depending on the 3 LPS molecules we studied. And there are hundreds of more. And you could probably combine that with the next proteins which come down here, which we haven't done yet. I mentioned that I should only briefly talk about TLR4, and I take this as a transition slide to the last example, and now from functional receptors, it'll be quick, from rather functional studies on 2 receptors, I go to many receptors at the same time, to networks of receptors. So both of these receptors I introduced, TLR4 and TNF-01, they act in a network. And I just put this very brief summary, which the left one is from Sun's review last year, Nature Reviews in Immunology, because this one discusses an interconnection between pattern recognition receptors. TLR4 is one of those, which is here, and the TNFR superfamily, so the 2 receptors I just presented. So they are tightly connected, already here at the level of the initial signaling hubs. And we have started to address this, but I'm not going to talk in detail about that, for TNFR1 activation together with Ursula Klingel's group. And what we propose is that we have to monitor more than 1 receptor. And I was very ambitious in saying that we have to monitor the whole receptome in a single cell. But what is important is in a single cell, it doesn't help us to look at 1 receptor in cell 1 and the other one in another cell. Because of this equilibrium and this network activity of these receptomes, we have to see them in the same cell. And if we were able to do that, we possibly would come up, or could come up with something like single molecule systems biology. Again, very, very ambitious. But let me just have it there. And why do I want to use it as a transition slide? Because, well, some 2-3 years ago, we have started with this network of proteins. It's a family together with an associated protein. As the EGFR family, which is EGFR and the 3 proteins HER2 to 4. And the associated receptor is the MET receptor. You could extend that, but we've initially restricted to these 5. And what you see here are some of the interactions or some of the important parameters we observe in the same cell. First one is expression level. They're very sensitive to expression level. The second one is all types of different interactions, ligand-dependent or independent, ubiquitination, endocytosis, and so on. And the third one is the relative expression of those receptors, because this also influences what kind of interactions and signaling hubs are initiated. And I show you very brief examples, indications that already at the network, at the receptor level, we have a network behavior. And these 3 examples are the monomeric and the dimeric MET receptor, the second one is the heterodimer, and the third one is the expression level. And it's really quick. It doesn't sound like it, but it's really quick. The first one is brings us to a live cell. We are now tracking the MET receptor in the live cell using a ligand which does not activate MET receptor. So we see the mobility of this receptor. And this is great data. I like this a lot. And well, to see a little bit more in this movie, we need some help for our eyes. So what I did is that I colored one of those MET receptors, a green circle whenever it's a single receptor, and when it found a partner, the circle turns red, and we have a co-locomotion of this receptor as a dimer. And then they dissociate again. And you can appreciate that this receptor, this single receptor, all sorts of looks for different partners. Some of those receptors meet multiple times, others stay together for a longer time. Let me restart this movie. And we can nicely follow that and extract the kinetics. This is the resting receptor. And the important thing why I like to discuss that is what we see here is a dynamic or a dynamic equilibrium between a monomer and a dimer. Right? So they associate, stay together, and then they dissociate. And this seems to be a general model for many receptors, like for TLR4 and others we've seen before. When we increase the expression level of MET receptor, many more of them are dimers. And then we activate the signaling pathway. So that's the first thing we observe. What we also can extract from these as movies are the diffusion properties. When this receptor is targeted with an activating ligand, for example, a growth factor, HGF, we see a drop in the diffusion. Internalization and all these kind of things happen. So that's additional information we have here. So this was a single MET receptor. Now let's link it to a second one. And I chose EGFR for that. Now we can track both EGFR and MET receptor in the same cell. And we can use both ligands, EGF and HGF, for the respective receptors. And what we see is that, well, it's not a surprise that EGF activates EGFR, but we also see that HGF seems to activate EGFR. So there's a cross-interaction. This is not really new, it's known in biochemistry. What was not really known is that also MET receptor is cross-activated by EGF. We can see that as well. But even more complicated, this cross-activation depends on the expression level. And we found that by looking at two different cell types, HeLa, which was a sort of the running horse for these experiments, but the breast cancer cell BT20. And we have a much stronger cross-activation of MET receptor in these BT20 cells. And this initially wasn't really clear to us, but then we looked at RNA-seq data, which you can translate into protein copy numbers. But we have super resolution methods in our hands, so we can also measure the expression levels of these receptors in single cells. And what we find is that MET receptor and the EGFR are approximately at the equal receptor expression level on the cell surface. That's important, only on the cell surface. Whereas in BT20 cells, we have a ratio of 7:1, quite different. So we cautiously contribute or attribute this behavior, this different behavior of crosstalk, to the relative expression level of these receptors and the dynamic equilibrium. So that's about it. I have the clinical paper, which I stopped because I would like to talk a minute about that, otherwise this will get too late. Let me just summarize it, what I've shown you. There's receptor imaging. We can do 2 or 3 receptors. We'd like to do many, many more. This has some technical requirements, which we are close to meet with new single molecule imaging technologies. I think it needs more than fixed cell imaging. We also, for functional aspects, need live cell imaging. And we need to sort of extract these kind of inter-receptor interactions, those network kind of interactions. And maybe we can build up a model, a single molecule model of how receptors act as a network in specific cells under specific conditions, and maybe also in response, for example, to drug treatment. That's about it. I'd love to thank my group, funding, and you all for your attention. Thank you.
Yeah, thank you too for this interesting